Good morning, class. Welcome to the biology class. This is class 11, and we are finished with the chapter 1 that deals with the general characteristics of the living organism, the naming of the organism, the binomial nomenclature, and the taxonomical hierarchy. I think all of you understood that topics. Now come to the second topic, that is a biological classification. And before that, let me remind you, please be ready with your book. And if you don't have the book, download the book so that it will be easy for you when you watch this videos. Okay, all of you stay safe, stay home. So let's start with the topic, that is a biological classification. In the last classes, we have seen that what is the need of classification, why they have been classified into plants and animals, and what are the different microorganisms are there. And in plants as well, we can see different group of plants. Some are with flowers, some are without flowers, and in animals we can see some are with a vertebral column and some are without vertebral column. So you can see a large varieties of biodiversity of animals and plants in the world, and how we can classify, and that is coming under the biological classification. So before starting this classification system, many scientists worked on the system of classification, and now what classification we follow is the final classification. But before that, many of them have tried with the different types of classification, and whenever any one scientist tried with a classification, there have been lots of advantages, there have been lots of disadvantages, and the other people who came in the way, they modified all these disadvantages and they try to improve the system of classification. So let's see one by one which are the different systems of classification we have been accepting, and how these organisms have been classified into different kingdoms. Okay, so let's one, that is a classification history. You know the picture, whose picture is this? This is a famous picture of our scientist Aristotle. Okay, and according to the Aristotle, he classified, so this is Aristotle, he classified the plants into trees, shrubs and herbs. Clear? So that was a very simple type of classification. And what about the animal? He classified the animals into two groups. One is those are having the red blood and those who do not have the red blood. Now, by listening to this Aristotle view, we might be thinking that what a foolish man is. He's not a foolish man, but he tried with the classification. Okay, so after this statement, the many other scientists started thinking how you can classify an animal depending upon red blood cells. Yeah, in the absence of red blood cells, so that is not a good type of classification. So they work for a better one. Okay, so this was first one, that is Aristotle classification, that is simply classified the plants into trees, shrubs and herbs, and animals into two groups, that is one who is having the red blood cell and one who don't have that red blood cell. Now let's see the next one. So the next one is by the Linnaeus classification, and you can see the picture of the Linnaeus. This is also a familiar picture for you. So this is Linnaeus who classified the whole group of living organisms into two kingdoms. Which are the two kingdoms? One is Plantae and other one is the Animalia. And we were using this classification still recently because we were not able to find out any better classification. So we were using, you have seen in the last chapter, we are classifying into two kingdoms. One is plant kingdom and other one is animal kingdom. Okay, so we were following this type of classification for a long time. And what is the disadvantage, or you can say what are the demerits of this system of classification? You can see here, he is not able, he cannot distinguish between eukaryotic and the prokaryotic organism. Okay, that is one demerit. And the second one, unicellular and the multicellular organism. And the third one, the photosynthetic and the non-photosynthetic organism. Photosynthetic, we know that they are the green algae, and the non-photosynthetic, they are the fungi, because we know fungi belong to heterotrophic organisms. Okay, so these were the demerits of the classification made by the Linnaeus. So what was the Linnaeus classification? He classified the whole group of the living organism into two categories. One is a Plantae and other one is the Animalia. But what was the demerits? He was not able to distinguish, he was not able to divide the animals, divide the plants depending upon the number of cell, the primitiveness of the cell, or whether they produce, whether they are able to do photosynthesis or whether they are not able to do photosynthesis. Okay, so these are the demerits, but we were following his classification still recently, but they are having some demerits. Now, what is the next one? The third option that we are going to study is about the five kingdom classification. I think you might have studied a little bit of this topic in your class 9 syllabus. And who is the proposed, who proposed the five kingdom classification? It is R.H. Whitaker. Okay, Whitaker's five kingdom classification. It is famous for the Whitaker's five kingdom classification. And I'm showing the picture here. The first one is Monera. So you can call it as Kingdom Monera, then Kingdom Protista, then Kingdom Fungi, then comes Kingdom Plantae, and finally the Animalia. This also is a better type of classification, but here also we can see there are lots of demerits he faced when he classified into the five kingdoms, because some of the animals like viruses and all were not be able to put into any of these five categories. Clear? So this is a five kingdom classification system. So the starting with the Monera, then comes the Protista, then comes the Fungi, then comes the Plantae, and then comes the Animalia. Now he is the person, R.H. Whitaker. So R.H. Whitaker proposed the five kingdom classification. He classified the whole animals into the Monera, that is the first one, then Protista, then Fungi, then the Plantae, and then Animalia. Okay, now what was his criteria for the classification? He considered the cell structure. Clear? Then comes the thallus organization. Then what is the mode of their nutrition, how they reproduce, and what is the phylogenetic relationship. What do you mean by the word phylogeny? Phylogeny, something stands for the evolutionary. Okay, how one organism is connected with the another organism, that is called as the evolutionary relationship, or you can call it as the phylogenetic relationship. So what are the criteria for R.H. Whitaker to classify into five kingdom classification? First one, they consider cell structure, then thallus organization, then mode of nutrition, then reproduction, and the phylogenetic relationship. Clear? So these were the criteria that he has used to classify the whole kingdom, the whole group of the living organisms into five kingdoms. The Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. Now, again, we are giving two headings like what are the advantages of five kingdom classification and what are the disadvantages of five kingdom classification. This one you have to study. This is an important question for you. So we can see what are the advantages of five kingdom classification. So first of all, what was lacking in the other classification, he rectified that. That is prokaryotes. Prokaryotes, they have a different cellular structure because they don't have what we can say proper cell wall. They don't have any of the cell organelles. They don't have a proper nucleus. Okay, so they have been kept under the different kingdom that is named as the Kingdom Monera. So the Kingdom Monera contains the prokaryotic organism. Now the second one, again he is rectifying the other classification that he kept unicellular and multicellular organisms in separate groups. Okay, then the third one, in that we have seen that the previous one we have seen that the fungi has been not separated. Okay, but here the fungi are placed in a separate kingdom because their mode of nutrition differ from the other plants, because we can't include the fungi with the plants, because we know that the plants are autotrophic organism but fungi are the heterotrophic organism. What is heterotrophic? Heterotrophic means the organism that depends upon other organism for their food and nutrition. Of course, that is the plants. They depend upon the plants for either directly or indirectly for the production, for the making of the food. Okay. Then the next one, the autotrophes and the heterotrophs have been placed in the separate groups. So this is another advantage. Then the five kingdom classification is more natural than the two kingdom classification. Then five kingdom is mainly based on the biocomposition, then morphology and phylogenetic, and thus it shows a series of the evolution. Okay, clear? I think it's clear for you. These are the advantages. What you can say actually, these are the rectification of the previous type of classification. So we can call it as these are the advantages of the RH Whittaker's 5 kingdom system of the classification. Now let's see the now come to the disadvantages. So what happened here? The unicellular algae is kept in the kingdom Protista, whereas algae like multicellular organisms are kept in the Plantae, and but similar organism must be put together. Clear? Now, second disadvantage is that there is a diversity in the kingdom Protista, because many of these Protista are very dissimilar organism, but they have been grouped into one. So this is another disadvantage. Then all organisms other than Monera, they are polyphyletic in origin. It's a very great shortcoming. Polyphyletic matlab they have been connected to different groups. Okay, poly means many groups, phyletic matlab it's like a phylogenetic evolution. So they have been connected to the many groups, and he is not able to show this one. And the next one, there is no place for the virus in the 5 kingdom classification. This is another disadvantages. We are not able to place this virus in any of these 5 kingdom of the RH Whittaker. Okay, and the such similar organisms are kept far away from each other. Okay, like unicellular and the multicellular algae. So that means even though they have been connected very much, they have been closely related with each other, but they have been kept in the different groups. Okay, so these are the disadvantages of the 5 kingdom class. You have to go through both of these. First one, what are the advantages, and second one, what are the disadvantages of the 5 kingdom classification. So we are going to discuss one by one what are the characteristics of the Monera, then Protista, then fungi, then Plantae, and then the Animalia. So this is the 5 kingdom class. Even though there are lots of demerits we have discussed, but we have to study what are the different characteristics, yeah, which organism is coming under which group. Yeah. So let's see the first kingdom, that is the Kingdom Monera. So before starting into the Kingdom Monera, let's see the general characteristics of all the kingdoms together. So here I have written here the kingdom. Okay. Then which are the kingdoms? Monera, then Protista, then fungi, then Plantae, and then the Animalia. So the characters have been given in the upper side, that is cell type, then nuclear type, then the what kind of cell wall, then what is the mode of nutrition, and what about the cell, whether they are unicellular, multicellular, like that. Okay. So the first one, Monera, so they are prokaryotic, and only the Monera is prokaryotic, and the rest of it is are the eukaryotes. Eukaryote, we know they have a proper cell wall, they have a proper cell membrane, and all the cell organelles are present. There is a proper nucleus. Now, what about the nuclear envelope? So Monera, it is absent, but in all others it is present. It's easy for you to study if you make a chart like this. Now, what about the cellulose? Monera, it is of non-cellulosic. Protista, present in some forms. The cell wall is present in some form. And in fungi, the cell wall is made up of a chemical called the chitin. And in Plantae, we know that the cell wall is mainly made up of cellulose and polysaccharides. And Animalia, we know the cell wall is absent. Okay. Then the mode of nutrition in Monera, it is autotrophic and some are heterotropic. And in Protista, photosynthetic or heterotrophs. And in fungi, they are the absorptive heterotrophs. That means they are depending upon the other organism for their food and nutrition. You can call it as many of them are saprophytes, because they obtain their nutrition from the dead and decaying organic materials. Okay. And Plantae, completely photosynthetic. And Animalia, again, they are heterotrophic organism. And in this heterotrophic, we can have the saprophytes and even the parasites also. Now, the cellular structure, if you see, Monera, they are absent. The multicellularity we are discussing, so they are absent. Then Protista, absent in most forms. And in fungi, present in most forms. And present in all forms in Plantae, as well as in Animalia, it is present in the all forms. So you have to learn this chart, so it will be easy when you come to each and every kingdom, so that you can identify what is the cell type, what is the nuclear envelope made up of, and whether they are having a nuclear envelope, and whether there is a presence of cell wall, and what is that cell wall made up of, whether the cell wall is absent. And you can see the mode of nutrition and even the cellular level, whether unicellular, multicellular. Okay, so this chart you have to learn. that will help you to study further. Okay. So let's come to the first one, that is called as a Kingdom Monera. So whenever you hear about the Kingdom Monera, it's actually made up of the bacteria. So the bacteria is the main member of this Kingdom Monera, and we know that they are a microorganism and they are the most abundant microorganism, and this bacteria can see everywhere. Okay, wherever you are sitting, in your air, in your atmosphere, inside your body, everywhere you can see the bacteria. And one of the important characteristics of this bacteria is that even this bacteria can be seen in places where other organisms cannot survive. Okay, like it can be seen in many extreme conditions, like you can see them in a hot spring where the temperature is very high, more than 100 degrees Celsius, and even you can see in the desert where the sandy soil again is very hot, and you can see in the snow where the temperature is very low, and even in the deep oceans or the trenches also we can see. Okay, and this bacteria, some of the bacteria are pathogenic, like they are called as a parasite, and some of them are even helpful also. Okay, don't think that always a bacteria is causing harm for us. You can think about a lactobacillus that is helping to convert the curd into the milk, sorry, the milk into the curd, and that is used as a microbe which is helpful for the human welfare. But some of them are pathogenic, of course, because they cause us different diseases. Okay, so this is the structure of a bacteria I'm showing here. You can see the different parts. There are lots of projections, cilia-like structure are there. There is a circular DNA and the cytoplasm, then bacteria flagellum. You can see the plasma membrane, you can see cell wall, there is a capsule, and they have the ribosomes. This is the structure of the bacteria. Now, depending upon the bacteria, we are going to classify into the Coccus bacteria, then comes the Bacilli, then comes the Spirilla, and then comes the Vibrio bacteria. Can you identify the shape of this bacteria, why they have been called so? The Coccus bacteria, you can see this Coccus. Can you see what is the shape? This is spherical in shape. And what about the Bacillus? They are the rod shape. What about the Spirillum? They are the spiral shape. And what about the Vibrio? They are the comma shape. So that's why the name has been given. Okay. So depending upon the shape, we are classifying the bacteria into Cocci, then Bacilli, then the Spirilla, and the Vibrio. You can easily see from this picture and you can identify them because their structures are like that. And some of these bacteria are autotrophic bacteria, and some of them, like, they can synthesize their own food material. And some of these bacteria are photosynthetic autotrophic, and some of them are chemosynthetic autotrophic. You know the difference. In photosynthetic, they are utilizing the sunlight, and in the chemosynthetic, they are utilizing the chemicals for the production of the energy. And the majority of the bacteria are the heterotrophs. Okay, that means they do not synthesize their food, but they depend upon the other organism, might be it is a dead organism, for obtaining their food material. Clear? So this is about the bacteria. Now, this Kingdom Monera, these are all by the Whitaker only. After that, many modifications have been made. So, the division of the Monera is divided into two groups. One is called as the archaebacteria and other one is called as the Eubacteria. There is a difference between these two bacteria. Let me tell you, the word archaebacteria means the bacteria that you can see in the extreme condition, or you can say in the harsh condition. And the word Eubacteria means that is a true bacteria. Okay, so that is a major difference from their name. Archae means they are being seen in the very extreme climate, yeah, extreme condition, and in the Eubacteria, they are the normal bacteria that is seen everywhere. So let's see the first one, that is the Archae. So archaebacteria are the group of the bacteria that you can see in a very harsh habitats. Okay, and how they can survive in the harsh habitat, because they have a very different cell wall structure, and this cell wall structure helps them to survive in the extreme condition. So I have given you the picture so that it's easy for you to identify. The first one I am showing you is the bacteria that can survive in extremely salty condition, And such bacteria you will call the name as the halophils. And this is the picture I am showing here of the halophils. So this is the place halo. What do you mean by the word halo? That means salt loving places. So these bacteria you can see in the places where there is a high concentration of the salt, and such bacteria you will call it as the halophils. Now the second group that you can see in the hot springs, yeah, you can call it as thermoacidophils, where the temperature is very high. This is a picture of a thermoacidophil, and you can see they can even this archaebacteria, they can survive in this harsh condition. Clear? Now the third one I'm showing here is a marshy area. Marshy area means it's like a wetland. Okay, always there will be the water there, and you can say कीचड़ जैसे. And these group of bacteria you generally call it as the methanogens. So this is a marshy area where you can see the methanogen bacteria. Okay, so these are the three extreme climates, and even in this extreme climates, this archaebacteria can be found. The archaebacteria will be able to survive. So this is also a picture that I'm showing here. This is a methanogen bacteria. You know that this is the cattle, this is a cow, and this methanogen bacteria are present even in the rumen. Yeah, rumen part of the cow. This is the intestinal part of the cow, and that is the exact place position is called as a rumen. And this rumen contains this methanogen bacteria, because this we know that the cattle's main food is they are taking lots of grass, and they contain the cell wall of this grass is having the cellulose, and to digest the cellulose this bacteria is needed, and this bacteria will help in the digestion of the cellulose, and with the help of this digestion of the cellulose they will help in the production of the methane gas, that is biogas. We know that we are taking the cow dung and we can help in the production of the biogas. So how it is possible? Because this cow dung contains the group of bacteria called the methanogen, and methanogen helps in the production of the methane gas, carbon dioxide and hydrogen sulfide, and this act as the biogas. So if you have been asked the question, what is the economic importance of archaebacteria? What's the question? What is the economic importance of archaebacteria? Then you can write this answer. They have been having the methanogen bacteria and they are helping in the production of the biogas. Clear? So I think this archaebacteria is clear for you. Now let's pass to the next one, that is the eubacteria. Okay, so already we told this eubacteria, they are called as the true bacteria because they have a normal cell wall, the rigid cell wall, and some of them are motile, and if they are motile, they have the presence of the flagella. In the first slide, you have seen the structure of the bacteria with the flagella. So some of them are motile and some of them are not motile. If they are motile, they contain the flagella. Okay. Now, some of these eubacteria, they are also called as cyanobacteria, yeah, blue-green algae. So they can fix the atmospheric nitrogen. And there are some special structure. You can see here, there is some special structure given here, and that is called as the heterocyst. With the help of the heterocyst, they can fix the atmospheric nitrogen. We know that the nitrogen is present in the atmosphere and they has to be fixed in the soil. Then only it can be absorbed by the terms of nitrite or the nitrate. So that has been done with the help of heterocyst by the eubacteria Nostoc and the Anabaena. Okay, and they also help in the nutrient cycling process of the nitrogen, phosphorus, iron and the sulfur. And some of these are the heterotrophic bacteria. The eubacteria are heterotrophic and they are mainly the decomposers. Okay, they are mainly the decomposers. Heterotrophic means they depend upon the other organism and most of them act as a decomposer. So these are the economic importance we can say. They act as a decomposers and they are helpful for the human welfare. That is like lactobacillus is there. They are helping in the conversion of the milk into the curd. And some of them are helping in the production of antibiotics. Okay, then some of them helping in fixing the nitrogen in the leguminous roots like the rhizobium bacteria that is present in the pea plant, in the roots of the pea plant. They help to fix the nitrogen. Okay, so these are the human welfare activities, like some of them are helping in the process of making milk into the curd. Some of them help in the production of antibiotics. Some of them help in the fixation of the nitrogen, that is especially in the leguminous plant. Okay, and many of them act as a decomposers, and most of the bacteria are the heterotrophic organisms. Clear? And some of them are pathogenic too. So which are the pathogenic one, or which are the diseases caused by these bacteria? They can cause cholera, they can cause typhoid, they can cause tetanus, and in the plants they can cause the citrus canker. So these are some of the diseases that can be caused by the bacteria. So here I am showing the picture where you can see these are the roots of a leguminous plant, the pea plant, where you can see the root nodules, and with this bacteria is present in the root nodules, and they help in the fixation of the atmospheric nitrogen. Okay, now how these bacteria reproduce? Some of them reproduce by the fission. This is a picture of the fission where you can see the single form. First, the nucleus is getting divided. Then after that, the cell wall is, the cytoplasm is getting divided, and you are getting the two daughter cells. And some of them, they reproduce by the production of the spores, and some of them, they adopt to a primitive method of just transferring the DNA from one bacterium to the another one. So these are the different modes of the reproduction that can be seen in the bacteria. Clear? I think these are clear for you. So we have learned only one kingdom today, that is the Kingdom Monera, which contains two categories. What is archaebacteria, and other one is the eubacteria. Archaebacteria is the one that you can see in the harsh condition, your extreme climatic condition, because of their cell wall condition. And the second group is the eubacteria, that is the true bacteria that you can see normally everywhere. And some of them are the photosynthetic, some of them are heterotrophic, some of them are chemosynthetic. Okay, and in the heterotrophic, they can see that some of them act as the decomposers, some of them are useful for the human beings, and some of them are pathogenic. Okay. There is one more there, that is mycoplasma. This mycoplasma, they completely lack a cell wall, and the most important thing is that they are the smallest living cell, and they can survive without the oxygen. Okay, so this important, you will learn in detail in the later classes what is mycoplasma, what is their structure and all. I'll say showing you the picture of the mycoplasma here. So they are the smallest living cell, and they can survive without the oxygen. That is the most important multiplication property of this mycoplasma. They can be grown anywhere, even they can survive without the oxygen. And these mycoplasma are pathogenic in the plants as well as in the animals. I think it's clear for you, the topic. So please go through all these topics along with your NCERT book. You can see all the pictures in your NCERT book. The pages are there. So we are finished with the Kingdom Monera. In the next class, we will start with the Kingdom Protista. So whenever you hear the word Kingdom Monera, now you understood that is comprised of the bacteria. And this is the classification of the RH Whittaker. There are lots of advantages and the disadvantages. So let's go to the next topic, Protista, in the next class. Hope you all understood. Make this topic very clear so that it will be easy for you to study for the next topic. Thank you.